finally, how can we determine, or how can we connect this type of graph in our heads with the type of graph that we talked about a couple minutes ago, our logarithmic growth graphs? Let's get rid of this stuff. So again, these graphs map the number of bacterial cells in a population. We're going to be drawing this type of graph on the next slide we look at. So keep this in mind. If the cells are in logarithmic growth, we are going to see a straight line. If they're not in logarithmic growth, then you wouldn't end up seeing a straight line, as long as it's graphed on this type of scale. So these types of graphs, and we see a couple of them in the textbook, show the growth rate on the y-axis. That's not what we were looking at before. We were looking at the actual number of bacterial cells. This is the rate, or how fast the population increases, or how fast the cells divide. And so in these types of graphs, we're actually comparing the rates over time. In other words, we're looking at the slope of this part of the growth curve, so the slope of log phase, and we're looking at the slope of this line. And again, looking at log there, this also shows log here, and these only show log phase. So if we zoom in here on the cytophile curve, it's showing that at these points, the growth rate is lower, right, than the peak. So this is the fastest growth rate on this particular curve, on this blue curve. And so what they're showing here is that at a pH of 1, the log phase is going to look like this. So the slope of the line is such that it's not very vertical. It's almost horizontal. Then if we compare that to the growth rate at a pH of 2, then we start to see a much higher growth rate, or a faster growth rate. And if you compare these two, this line has a greater slope to it. So it's becoming less horizontal and more vertical. Then finally, if we look at a pH of about 3.5, if we look at the maximum point here, then the slope of the exponential growth curve, or the log phase, is as steep is steeper than the other two graphs that we saw here. So this is the fastest growth rate, which is shown here. Now these, as you might be gathering at this point, these types of curves, or these types of graphs, take a lot of data, because you need to be growing bacteria through log phase. So in other words, you have to get past lag phase and go into log phase for one population of bacteria at 1 pH, and then you need to do that all over again for another population of bacteria at another pH. So another thing to understand is that you're not using the same population of bacteria and putting it at different pHs. You're using the same type of bacteria, but we're not going to use the same exact bacterial cells to map this out. And so you could apply this kind of schema to all of these different curves on this graph, as well as this graph here. And what the takeaway is from these is that the growth rate of bacteria is going to be higher, of different types of bacteria that is going to be higher in certain conditions. And some bacteria will divide the fastest at cold temperatures, while other ones are going to divide faster at a kind of a room temperature or a moderate temperature. Some bacteria do best in hot springs or in super hot springs. So this is not to say that all bacteria will do best at, a hot temperature, but certain species of bacteria will grow best at a certain temperature. And so if you had one specific bacteria that you needed to grow a lot of, you might consult something like this with the temperature for that bacteria, and then you would also want to consult a graph like this showing the peak growth rate or the optimal pH where you get the highest growth rate. And so again, this is about optimizing the conditions to grow a specific bacteria.